everybody, welcome back. Today we're going to be talking about DNA. First of all, can someone help me remember where is it that we find DNA in our cells? What organelle are we going to find DNA in? That's right, it's found in the nucleus of each and every cell. Check out this diagram and please make sure you label it on your green note sheet. Look at how all of the DNA that is in the nucleus of each cell is actually wrapped up into that X shape called a chromosome. And we are going to be exploring this a lot more soon, but it's very important to know some of this vocabulary. So please label this diagram in your notes right now. Got it? Great. If you need another minute, just hit pause. DNA is short for deoxyribonucleic acid. So all of those parts mean something. It has to do with the different substances that make up the actual strand of DNA, the chemistry of DNA. So they all stand for something. So deoxyribonucleic acid. This substance coils up to make your chromosomes, just like you see over here, that X shape. So all of that DNA coils up to make chromosomes, and this is especially important during cell division, when cells are getting ready to make more cells. When the DNA is organized in that shape, it's able to divide more cleanly and more quickly. Each chromosome contains an enormous amount of DNA. DNA, when uncoiled from that chromosome, kind of looks like a twisted ladder. This shape is called a double helix. The names of the scientists that discovered this shape were James Watson and Francis Crick. However, these guys didn't do all of this completely alone. They did build their DNA model on the theories of many other scientists who studied genetics, but these are the guys who we get the most credit for and give the most credit for discovering the double helix shape, which is that twisted ladder. So if you were to take just a regular step ladder and give it a twist, that's what we call a twisted ladder or a double helix. Here we go. Check out these DNA parts. Every part of DNA is a different sort of substance, a different chemical. Each part is called a nucleotide because of the nucleic acid that is a part of it. You notice how you see this same picture at the bottom of your paper. The sides of the ladder are two substances that always alternate. They are always a phosphate and a sugar. So a deoxyribose sugar and a phosphate. So up and down the ladders go sugar, phosphate, sugar, phosphate, sugar, phosphate. Notice how attached to every single deoxyribose sugar are these different rungs, and this is where the base pairs match up. The four different bases are adenine, thymine, cytosine, and guanine. These are the names of the different nucleotide bases that make up the rungs of the ladder, these different chemicals. Look at the picture on the bottom of your notes. Please take a minute to finish labeling that strand using the same shapes that you see here on the paper. Got it? Great. So all of these pieces fit together in a certain way, and then we read the DNA strand, the different parts, we read them like a code, so we can figure out what each part of the DNA code means by reading the different letters. To give you a better idea, let's go to an Amoeba Sisters video.
So DNA stands for deoxyribonucleic acid. That's what the Na part is, nucleic acid. And that deoxyribo, that's for deoxyribose, which is a sugar. So nucleotides have three parts, and one of them is a sugar called deoxyribose. The next thing is a phosphate. The phosphate is involved with the sugar in helping hold the DNA structure together. We sometimes call DNA as having a sugar phosphate backbone. But the most important part of the nucleotide is the base, because the bases actually code for traits. So as far as the bases go, again, those are the important parts that control your traits. They really directly code for your traits. There are 4 bases in DNA, and a lot of times they'll just use the initials, A, T, G, and C. The A is for adenine, the T is for thymine, the G is for guanine, and the C is for cytosine. There's a really nice little saying that can help you remember which one goes with which, because these bases actually pair in a correct pattern. And if you don't have them paired correctly, or maybe they get mismatched, that's actually what we call a mutation, which we'll get to a little bit later. But here's the little way you can remember it. Apples in the tree. That tells you that A for apples, T for tree, A and T always go together. And then the other little verse is cars in the garage. That can help you remember that the base C always goes with the base G. And so you kind of know how they match. And DNA pairs this way. DNA has 2 strands. So there's nucleotides running up one side, and there's nucleotides running up the other side. The bases are what pair in the middle. They're usually held together by hydrogen bonds. The DNA is also twisted in something we call a double helix shape. And so as the strands of the DNA is twisted around, the bases should be all in the center. The sugar and phosphate make up the sides. So let's review what we talked about today. We talked about the importance of DNA, that it's found in the nucleus of cells, that all of your cells, every one of them, they contain all of.
Awesome. Thanks, Amoeba Sisters. Okay, so now that we know the basic structure of DNA, let's talk about when it's copying itself, because this is extremely important. So whenever your cells are undergoing mitosis, you want an exact copy of whatever is in one cell, you want it to go into the next one. There are 2 special ways cells divide. I just mentioned mitosis. Well, we'll learn about these processes next, but in both these ways that cells copy one another, the genetic material must be copied first. So here's how it happens. Here's how DNA makes a copy of itself. Check out at the bottom how that ladder seems to actually unzip. The DNA ladder unzips down the center, separating all those matching base pairs. So, oh no, the apple in the tree and the car in the garage, they're pulled away from each other. A new piece, though, comes in to replace the old piece on each side of the ladder. So you can see down here at the bottom how as the ladder unzips, all of a sudden the new pieces come into place and create 2 identical ladders. Pretty neat.
One of the most beautiful things about the structure of DNA is how it replicates. DNA replication is how the DNA makes an exact copy of itself, which it needs to do before the cell can divide. First, DNA helicase unzips the DNA by breaking the hydrogen bonds between the bases. Then, DNA polymerase adds complementary nucleotides to the correct bases in the original DNA strands and connects them. The complementary strands always match A with T, C with G. So if you know one strand, you can predict the other. If the original strand has these letters, the complementary strand will match T with A and C with G. It turns out that DNA replication happens smoothly and continuously on one strand, but in small broken sections in the opposite direction on the other strand. The continuous strand is called the leading strand, and the one built in sections is the lagging strand. The reason for this method of replication is that the bases must be added in only one orientation, sort of like the way Lego bricks have to be linked together in only one orientation. The lagging strand sections are connected with an enzyme called DNA ligase, and then the copy is complete. Errors in DNA are very rare, only about 1 in every billion base pairs. That's incredibly efficient, and it's a good thing that it is. Mutations can be harmful or even fatal. The enzyme that checks base pairs and repairs mistakes is DNA polymerase. It's the same enzyme that links DNA bases together during replication.
Cool. So, now we know how DNA is copied. But once DNA is copied, remember DNA has to actually be read and interpreted to be able to build you who you are. Do you remember the name of the organelle that actually reads the DNA? We practiced it during our cells unit. Yeah, remember him? It's the ribosome. That's the organelle that reads the DNA code. Reading DNA is like reading a book, but believe it or not, there's only 4 letters in the alphabet. Remember A, G, C, and T? A and T are always paired together, C and G. Apple in the tree, car in the garage. These letters are grouped into groups of 3, just like we practice when we practice being ribosomes. All 3 letters code together to group for a specific amino acid. Remember when we used this wheel? Yeah, for example, every time the bases A, G, and C line up, that codes for the amino acid serine. So the ribosome reads that DNA code to actually build the proteins that make you who you are. Here you can see the ribosome back here at work building a protein chain made from the DNA code. The amino acids link together to make proteins that make you you. Your hair color, your eye color, everything about you is in your DNA code. The DNA is just the instruction manual to build the protein that actually does the work. But sometimes there are little errors or changes in the genetic code. Uh-oh. Mistakes do happen, even though they could be even just 1 in a billion base pairs. Think of how many base pairs there are. Mistakes can happen. These mistakes are called mutations. Mutations can be caused by maybe a base pair being left out, an extra one added, or even one just being put backwards. Check out this chart right here. This chart shows you making blood cells, the code that your body has for making blood cells. Here you can see that glutamic acid is normally in this space to make your blood cells the shape that they are, like squashed donuts. But what if valine were to replace glutamic acid? What if the A and T were just to be reversed? That one error in the code would create a new acid, which then creates a whole new protein, and now the red blood cells instead are a completely different shape, and this can cause a very fatal condition called sickle cell anemia. So what is it that causes mutations? A mutation, if it happens in one of the sex cells, it can actually be passed from one generation to the next. But what else causes them? Mutagens, like too much radiation from X-rays or being exposed to UV rays, can be damaging. Chemicals like asbestos and cigarette smoke, all of these things are called mutagens. Just because you're exposed to them doesn't mean you'll necessarily have a mutation, but mutations are directly linked to being exposed to these. Well, that's all I have for this time. Thank you for watching and learning the basics of DNA.